Good afternoon, everybody. We are here today to the webinar from the Caribbean Hub. Today, we will be listening to Dr. Melanie Bordeaux from the Foundation of Nicafrance. Melanie obtained her doctor's in chemistry in 2012 in Montpellier, France, after completing her doctoral studies in investigations, postdoctoral investigations in biochemistry and in biotechnology in the United States. Since 2015, she has worked in the harvest of coffee in Cumplida in Matagalpa, Nicaragua, where she is the director of the foundation of Nicafrance. Her areas of interest include the uh, genetics of coffee and of cacao, and the agronomy and the systems for the agro forestry and methods of post-harvest. We are very thankful to have Miss Melanie, Dr. Melanie, with us today. On the 26th of July, we will be having another subject matter. The International Symposium of Cacao. Um, we hope to be completed with all of these workshops in order to learn more. I apologize, I can barely hear the speaker. So now we are going to proceed with Miss Melanie Bordeaux, Dr. Melanie Bordeaux, and she will continue with her presentation. Thank you for that wonderful introduction. Good day to everybody. And I'm calling from I am here today from Nicafrance. I'm having a little bit of technical difficulties, but I hope that you will be able to hear and understand fully all that is being presented today. Can you all see my screen? Screen is being uploaded and shared. We are all going to turn off our screen and I will also just to assist in the by turning off the video, we can assist in perhaps um, less wear on the system. I don't know if I can see you right now, but just some technical difficulties. We are going to resolve these technical difficulties shortly. She is going to begin by giving a brief uh, introduction. Today's subject is the use of the SNP markers for the genetic and traceable print of cacao. This project commenced in 2018. And Nicafrance is a combination or collaboration between Nicaragua and France is the organization's name. We joined forces with exporters from Nicaragua by cross-sectioning or selecting several different trees in the Nicaragua area in order to be able to put some genetic markers and traceable markers of the cacao in that region. And what we are hoping to do is to take this methodology and to be able to apply it to other regions. Although fundraising or funds were a little limited, this has been applied well in other regions already. Okay, there. Now we have the shared screen. And for those of you who are producers or harvesters of cacao, sometimes our expectation is very different than our reality. You can see a beautiful image of what our expectation is, and then the reality is quite different. And when we see the catalogs for cacao, we're inspired by certain images. the images that were shown in a catalog are very beautiful, and I'm not doubting that in some regions it can look that way. But in reality, the production or the yield gap is much less than what our expectations can be. The difference or the yield gap could be very different than what our expectation was. A difference between 3,000 kilos to as low as 1,000 kilos. So the gap in the yield is the difference between the reachable yield and the real yield for agricultures. For example, as we look at these different um, slides here, in Ghana, for example, the yield gap is 50 to 82%. In the case of Latin America, these are the catalogs, the cloning catalogs, and we can see that they're actually very good. This is a cross section of cacao in Honduras, Guatemala, Nicaragua, and El Salvador. For example, in the case of Honduras, this is the FAO statistical data where you can see the production of cacao, again, the expectation versus reality. And this is the measurement is kilograms by hectare. What can we see here in this red column? That all of the countries that are producing cacao are much lower. Guatemala and Dominican Republic are special cases, however, and it is very probable that these have been overestimated, this data. But we are definitely overall very under the expectation. So why this yield gap? So we're going to explain now the possibilities for the gap in this yield. There are different factors that cause this gap. between the expectation of what is planted and what is harvested. We could have um, ergonomic practices, infrastructure technology, environmental factors, social and organizational factors, and economic factors. But today we're going to focus on how to make better or better the harvest. These ergonomic practices can be bettered by implementing different varieties, fertilization, control of P&E. P&E is pests and disease, to pruning and making it more compatible. She is naming several pests that enjoy the cacao plants. The maintenance and pruning is also a large factor. Next, please. So the question is, how can we make this better? How do you make it better? We can make it better by entering into more knowledge with regard to the genetic material. In other words, we need to have a better understanding. And how can we do that? We can better the genetic, we can better the genetics and productivity by using more resistant variations, better arrangements, clones, and better quality. And when we understand these, obviously we can um, increase our efforts in the area. And in different countries, in different regions, we might have different pests, we might have different soils, we might have um, different elements that can affect. And so we do need to conserve the diversity, the genetic diversity and the sustainability, and therefore the resilience. Also, we need to authenticate and certify um, or make it more traceable to order to certify and create a greater value in the market. So the baseline is, what do we know about our material? The origin of the material is genetic. Seeds, reproductions, vegetation, and grafting, and somatic embryogenesis. The example that we have given of Honduras is we needed to go and visit the actual individuals, agricultures, that to see if they knew what elements affect impact for the genetic elements of their own crop. And in the adding of clonings, we can increase, but there has to be knowledge about this. There have been problems in identifying let me go to the next one. The markers, the SNP markers, are molecular markers. They are called single nucleotide polymorphism. They serve as, so how can we know our genetic material? The genome sequences of the cacao has helped us with this discovering of the single nucleotide polymorphism. They are helpful for the making of germplasma due to the low cost and the facilitation, automatic facilitation. These can be used to find the differences and to comprehend the diversity of the genes inside of that particular population by tracing its origin and its genetic characteristics. Next, please. In the case of selecting SNP markers reflecting the population origin for cacao, which is called Theobroma cacao germplasma identification. This is the SNP list in order to identify the germplasma of cacao. Next, please. Let's see. So in practice, this is how it looks. This is the cooperative, the different production houses of cacao in the cloning garden. We would collect the leaf samples and we would prepare them. Then we would extract their DNA, place them through the SNP sequence until we have an interpretation of a diverse genetic study and a base for the data, the genetic data, and from which to conform. Of course, in the country in which you are performing this, you would need to have a laboratory that can select and put into a sequence the extraction of the DNA. It does take time, however, but we're only trying to look at the SNP sequencing. And after receiving the genetic data or the base of genetic data, then you can proceed to bringing it into conformity. The collection of samples of the vegetative material for the extraction of the DNA. So various commercialized variety nationwide. The objective is to create a base of data for the traceable system. And it shows here in the images that it is collected a total of 42 samples of in from the clonal gardens. One in a different area and three in another area in Nicalizo. The art that the We collect all of these different samples from the different regions. These are the different samples that were collected in the different zones or production zones of cacao in Nicaragua. So the collection of the material, the vegetative material, they are collected in Ritter Sport Rancho Grande, and we interview the farmer. But so we don't just only collect the leaves, we also interview the farmer. And we take it to on the right-hand side, we have a farmer that wanted to join this union. So in Finca El Nuevo Amanecer, the farmer Jose Barrera, that's him there. He chose the seeds of a small fruit. It's very different. The characteristics of the special characteristics of this particular cacao tree is is known as monkey cacao. The extraction of DNA from the leaves. The picture at the very bottom is the is a very expensive machine that actually provides the sequencing of the DNA. But they are very specialized, and this machine assists in that, very specialized in reading the DNA of the cacao. This is the result of the sequencing of the DNA. This is the data that is interpreted. This is what we receive. The genetic database of the single nucleotide polymorphism, which is SNP. It gives us basically a print, a genetic fingerprint or footprint of the different genetic markers. So these statistic tools are very helpful to us. And so the geographic and genetic population differentiation of the Amazonian chocolate tree or the cacao tree. There's differentiations in the genetic groupings. 10 groups defined for by Montemayor and Company or ETL in 2008. We have the different clusters also that have been sampled in the different areas. But down here, there is more precise. In this slide, you will see the precision, the analysis by the principal components. By taking the different clustral samplings and the different trees, we're able to make the comparison. Now, we also do have to look for the hybrid trees, which are not easily identified. The genetic composition of the trees that have been taking samples in Nicaragua, these were the results. The genetic composition is very complex. 33 trees have more than 50% of their genetic genes that pertain to the same grouping. And 22 trees have their origins in more than 3 different groups. 40% of the trees have Creole sap or natural to their origin. It's 40%. The 22 trees that have origins in more than 3 different groups are preferred. That's very good because they are in yellow is the Creole. But the bands that are completely yellow, those are the trees or the areas the tree samplings that are completely Creole or original to the area. In Nicaragua, we've been able to outline the different groups. All the groups are represented here, but the groups more represented are the Amilionado or the Criollo IMC. Criollo meaning original or native. The white seed, the origin, again, continuing for the white seeds, the white seed does not necessarily mean criollo, as it is defined in the genetic groupings by Motamayor. You see the little stars on the images here? These seeds are totally white, but as you can see, there's very little identification by the yellow color of the bar, which means natural, original to the place, or criollo. So this demonstrates very well that the white seed does not necessarily mean that it is an original or native tree. Could be other factors involved. In the cross-sectioning or the selection of elite trees by the farmers, there are four criteria for this selection: the white seed, the resistance to plagues and diseases, their productivity, and whether the cacao is native. You can see here in the graphing area where the white sees the resistance and productivity cross section. Of course, the ideal cacao would be the native cacao. So this graph shows us the productivity, the tolerance um, to the monilla, as well as the which has the white seeds. So in here, in Nicaragua, this is the situation in Nicaragua. In Honduras, This is the study that was done in Honduras of the genetic diversity and the patronage in the farmer varieties of cacao. And in Puerto Rico as well. As you can see, Miss Stephanie Cole is the person who was, you can look for this diverse study in Puerto Rico by Stephanie Cosme Reyes. She analyzed in collaboration with, next please. In the case of Trinidad and Tobago, I'm sorry, I'm having difficulty hearing the speaker. This This slide is showing the International Symposium on the cocoa research, as you can well read in 2017, the new insight of genetic diversity and the genotype identification using the SNP gene-based markers. The conclusions of Nicaragua, and for what is this, why would we use, what use is this genetic analysis to us? The use is to be able to construct a national system of traceability of the plants, the cacao plant, in order to uh, organize the information and promote a finer cacao in Nicaragua and the development of a national brand. To also construct a database for the genetic printing of the cacao in Nicaragua and to determine the diversity, the genetic diversity, which is of the cacao that is actually in Nicaragua. In addition, it was to generate major, to generate better conditions for the investment and financing to contribute to the productivity of the cocoa growers in Nicaragua and to create conditions that are favorable to the registry of the cloning gardens and the farms of cacao to fortify in long term the confidence in these farmers in their material genetic product and the exportation of cacao, Nicaragua's cacao. These are the conclusions of the SNP as a base for a genetic printing. The SNP technique can be used to validate the genetic material in the gardens and in the farms of the cloning gardens and in the farms. The SNP also serves as an implementation of the certification and traceability of the genetic cacao material, making a system of certification. The traceability of these plants of cacao can guarantee productivity and compliance with those international standards of quality. The composition and the genetic unique ex the genetic composition, the unique and genetic composition explain the exclusive profile of the cacao in each of these countries. In addition, In addition, it would assist in controlling the dissemination of the genetic material and the gleaning of better sections of the cloning to increase the control and the compatibility in between the clones and to increase the production and the quality. I want to thank I want to thank you all for listening and your interest in here in this presentation. And I would like to open for any questions. You have there my 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 email that you can also forward any questions that you might have to me. There are several questions that are going to be asked here. We can also use your email, and then later on you might be able then to sorry, did not hear the question. so sorry. I can't, there's another question, but Mr. Luis, is it possible to associate the markers with their place of origin? There's various ways of doing it, and I've done a lot of work with that. What do we do with the four trees, the native trees that were found, the Creoles? It's a very good question. It's not just the Creole, the native trees, because all of them have a tremendous uh, eh, uh, benefits. So where do those Creoles come from? And other studies are being done to find that. Every origin or every native tree, every tree is important to this study. And what you're leading to is the conservation of these particular trees. The superb quality of the Creole here, especially in Nicaragua, because it increases the flavor and but when you ask the actual farmer, you would learn more about their particular uh, harvest. There's so many elements that affect a crop. So, but obviously in this presentation, it was only to give a summary of everything, but all those things can go up, can be delved into more, certainly. So obviously there's a plan to better the quality in all of the trees. The presentation will be sent to all who have participated here, and everyone will receive the actual presentation. And the webinar will be placed on a YouTube channel as well, with the translation and the interpretation. I know a lot of people are especially excited to know about the studies that were done in their countries, but there's a lot of studies going on. Thank you then, Melanie, for your time. Congratulations to you all in your work. And we certainly learned a lot today with regard to the work that you are doing. Thank you. Um, I hope that thank you again also, Melanie. I hope that each of you were able to comprehend and also to enjoy this presentation. In 2 weeks, we will have our last seminar with regard to this entire symposium on the cacao growers. Thank you to all of our sign language interpreters and interpreters as well.